Okay, so by now, you might be wondering how can the three-dimensional structure of a protein be determined? Here are some ways they do it. First, you can do X-ray crystallography. No? So this requires a perfect protein crystals. No? That is, one in which all individual protein molecules have the same three-dimensional structure and orientation. Note that you can only grow this kind of crystal from proteins of very high purity. Now, what happens if you were able to crystallize your protein? You will expose it na to a beam of X-rays, no, which will give a series of diffraction patterns. Then finally, information on molecular coordinates are extracted via mathematical analysis called Fourier series. So shown here is an example of an X-ray data. Next, you can do a 2D NMR. Okay, here, large collection of data are subjected to the same mathematical analysis as that of your X-ray crystallography, no, your Fourier series. Okay, and it uses protein samples in aqueous solution instead of crystals. Now, this method ultimately depends on the distance between hydrogen atoms present sa protein mo. No, so shown here is an example of NMR data. Okay, finally, we have here the quaternary structure of proteins. So this is the final structure of proteins. Note that, note however that not all proteins have quaternary structures. Unlike with the what do you call this, with the first three levels which are always present in all proteins. Now, so this level of structure is only what do you call this, is only present to proteins that have different subunits. No? So you can have here dimers, trimers, etc. And that each subunits, okay, are held together mainly by hydrophobic interactions. No? So also by electrostatic interactions and H bonding. Again, note here that the quaternary structure is only applicable for multi-subunit or multimeric proteins. Okay, now let's discuss about myoglobin and hemoglobin. Okay, at the onset, note here that these two are globular proteins and are heme proteins. Okay, meaning they consist an iron-ion coordinated to a protoporphyrin ring. Protoporphyrin, no, which is the non-protein portion of both of your myoglobin and hemoglobin, and that these two proteins are responsible in maintaining the supply of oxygen, which is vital for oxidative phosphorylation. Okay, myoglobin has a single polypeptide chain. No? So this is a monomeric protein. Now, as the name suggests, okay, myo, which translates to muscle, and globin, which translates to protein, okay, we can say that this is found in muscles. Okay, this serves as a storage protein, no, which reserves oxygen that the body can use in times of oxygen deprivation. Okay, by the way, myoglobin consists of a single heme group, no? So, yun, kaya nga siya monomeric. So, some trivia here, myoglobin is actually the first protein for which the complete tertiary structure was determined using XRD. Okay, so this was done by John Kendrew and Max Perutz, no, for which they won the 1962 Nobel Prize in Chemistry. Okay, hemoglobin on the other hand is a tetrameric protein, no, found in erythrocytes or your RBCs, no, which is responsible for the hemoglobin, no, is responsible for the transportation of oxygen to the tissues in the body, okay, and returns carbon dioxide and protons to the lungs. Yes, you heard that right. protons. Now, we'll discuss this under Bohr effect later. Hemoglobin here, okay, consists of 4 heme groups, no? So, one for each monomer. By the way, this is your protein. No, this is your hemoglobin. So, yeah. Okay, it is important to note here that these are two substances that is responsible for the capability of these proteins to transport and store oxygen. I'm referring to to the heme group. No, so focus your attention on the figure. One of those substance is your heme. No, so this is a cyclic tetrapyrrole linked by methylene bridges, and the other is your ferrous ion, which is at the center of the planar ring. No, so you have here your ferrous ion complex to the heme group. Okay, so some trivia here. What happens if, for example, your ferrous ion is oxidized to ferric ion? Of course, okay, the obvious answer would be your protein will lose its biological activity. But how does it happen? Okay, okay. So when ferric ion is the one bonded to your heme group, you call that your methemoglobin. Now, which is a form of hemoglobin. No? So note that this type of hemoglobin can also carry oxygen, yes. But the problem is it cannot release it effectively to the tissues, no, that needs it. So this results in a decreased availability of oxygen to the tissues. So therefore, increased concentration of methemoglobin in the system would further decrease the availability of oxygen to the tissues and would render the afflicted person to have a chocolate colored blood, no. And in addition, okay, when methemoglobin concentration is becomes higher than 1%, the person would exhibit a blue skin color. Now, so this is a disorder called methemoglobinemia. So again, methemoglobinemia. Okay, so this is the structure of your myoglobin. No? So again, note here that myoglobin can only bind one oxygen molecule because it only contains one heme group. No? So shown here in red is your heme group, no? Which is located in your hydrophobic pocket. So your myoglobin consists of 8 regions of alpha helices, okay, and no regions of beta pleated sheets. Okay, so the most polar side chains here is on the surface, and the non-polar side chains are folded to the interior. No, so this protein consists of again a single polypeptide chain, okay, with 153 amino acid residues, no, and a prosthetic group, which is your again nga heme. Okay, so note here that there are two spherical histidine there are two special histidine side chains present near the heme group, no, of myoglobin and also hemoglobin. No? So these two histidine residues assist in oxygen binding. No? Let's discuss this later when we're under the hemoglobin structure na. Okay. So this one naman is the structure of hemoglobin. And if you still remember earlier, we mentioned that there are 4 monomers present in hemoglobin. No? So each monomer is shown in different colors, and same with myoglobin, no? Shown here in red is the heme group. So since hemoglobin is a tetramer and each monomer contains one heme group, it follows that it can bind up to 4 oxygen molecules. Okay. Notice in the structure that there are alpha and beta regions, now as compared with the myoglobin, no, which only contains alpha helices region. So it means that hemoglobin contains 2 alpha chains with 141 amino acid residues each, and 2 beta chains with 153 amino acid residues each. No? So hemoglobin is represented by your Greek notation or Greek letter alpha 2, beta 2. By the way, you also have here pala yung fetal hemoglobin or fetal. hemoglobin, denoted as alpha 2 gamma 2. And the difference between the fetal and adult yung alpha to beta 2 is that the latter binds to the oxygen more strongly, allowing the transfer of oxygen from mother to fetus. Later kasi you'll see that in adult hemoglobin, lesser yung affinity niya with oxygen. Okay, note here that since hemoglobin contains different subunits or monomers, it follows that it exhibit quaternary structure. There. Because again, remember, a quaternary structure is only present in your multimeric proteins. Now, this level of structure provides additional properties to hemoglobin which are absent from the monomeric myoglobin. Remember, we said that hemoglobin can bind up to 4 oxygen or 4 oxygens. Note here that this protein exhibits positive cooperativity or tawag ng iba, cooperative binding. So, this means that when one oxygen is already bound to a heme, it becomes easier for the next oxygen to bind to the other heme groups from different monomers. So, the presence of oxygen in one of the heme group increases the affinity of other hemes to oxygen. Yung mga monomers mo dito, para silang magfe-friends. Okay? So, kung ano yung meron sa isa, yun din yung meron sa lahat. Parang sabi nila, uy, meron nang nakadikit sa aking oxygen, magkuha na din kayo ng mga oxygen nyo. So, ganon. It's like peer pressure. So, by the way, cooperative binding is also applicable to other molecules that can bind to the heme group. Okay, now let's discuss about the binding of oxygen to both myoglobin and hemoglobin. Okay, so remember here that your heme group contains iron in its ferrous form, in the center, coordinated in the 4 nitrogen bonds in the imidazole ring. If you still remember, okay, from our previous video, the amino acid which contains imidazole ring is histidine. Now, from earlier, we mentioned that there are 2 special histidine residues present in both myoglobin and hemoglobin. So, ito na yon. No? So, we'll discuss its importance later. Your iron here also binds also bind oxygen molecules. Now, so we can say that one of the axial position of your iron is the binding site of oxygen. Remember here that the heme group adopts a planar configuration. So, planar yung nakikita nyo sa structure. Okay, so if you think of the structure of oxygen, okay, you have here 1 pi bond, 1 sigma bond, and you also have your lone pairs. Now, ito yung molecule na dapat mag-bind sa heme group ideally. However, there are other competitive ligands that want to bind to your iron. For example, your carbon monoxide, CO, okay, we know very well that this is poison poison siya, and at the same time silent killer, silent killer to eh. So it doesn't have a smell. So you don't know if you're inhaling it na. Basta-basta ka nilang tutumba. Let's see why. If you look at the structure of carbon monoxide, okay, you'll notice here that it has a very similar structure with oxygen. And you will also notice here that it has a net neutral charge. Pero wag kayong magpapaloko dyan. Kasi if you compute for the formal charge of each atoms, you'll see that, okay, oxygen here will bear a positive 1 charge, which is not our concern, by the way, and carbon here bears a negative 1 charge. Okay, remember, positive charge attracts negative charge. But because of that negative charge, ulit nga, so remember that positive charge attracts negative charge. So meron kang negative charge kay carbon monoxide and meron kang positive charge kay iron. Now, pag nag-bond sila, magkakaroon ka ng problema because of that negative charge, no? So, dahil nga may negative charge siya, it doesn't want to leave the heme group. Now, that's a problem because since there's no oxygen being transported to your cells, okay, the oxidative phosphorylation will shut down since oxygen nga is important here because as you will know later, no, oxygen is the final electron acceptor. And later, you'll also know that this process produces the bulk amount of ATP needed by the body, okay, to function and to maintain homeostasis. Now, so this is the reason why carbon monoxide is poisonous, not just to us, but to any living organism that utilizes aerobic metabolism. Okay. By the way, note here that carbon monoxide has 250 times stronger affinity for the heme group as compared with oxygen. Which means that if a carbon monoxide gets bound to heme, it's gonna be very hard for an oxygen to kick it out. Okay. So makikita mo dito si hemoglobin, check niya si carbon monoxide, which is hindi niya dapat gawin. Okay. Another similar structure here would be cyanide. Okay, which also binds competitively to heme. Now, aside from this, it also binds to the complex 4 in oxidative phosphorylation. No? So, more of this later no, as we reach the said topic. So, makikita rin natin here that cyanide is poisonous. Again, you can see here that the binding site for oxygen is on the axial position of your iron. Now, this is the importance of the histidine residues near the heme group of myoglobin and hemoglobin. Okay, so the histidine residue bonded okay, to the axial position opposite to the binding site of oxygen is histidine F8 or the proximal histidine. Okay, by the way, the notation his F8 means that histidine is the 8th residue in helical segment F. So basically, it tells you about the position of histidine. Now, proximal histidine here strengthens the iron imidazole bond. Now, on the other hand, the other histidine residue is in the interior of the molecule is histidine E7 or his E7 or the distal histidine. Okay, so distal histidine here means that this histidine residue is located far from the heme group. But remember, kaya nandyan yan sa taas ng binding site is because nagfold yung protein mo. So same notation here. Histidine E7 means that histidine is the 7th residue in helical segment E. Now, distal histidine here is not bonded to the iron or to any part of the hydrophobic pocket. Bonded, okay, to the heme group. Note that distal histidine serves as a gate that opens and closes as oxygen enters the hydrophobic pocket and binds to the heme group. Okay, so I'll just share with you that I am very fascinated with the design of this because this is very critical in transporting oxygen. Okay, so now remember the function of myoglobin is to carry oxygen in muscle tissue. And hemoglobin naman transports oxygen to the tissues. Okay, different parts of the body. Now note here that the oxygen bound to the heme group should be easily released to the tissues that need it, no? Kasi of course you're gonna have a problem if mahirap i-release yung oxygen na need ng mga tissues because again, oxidative phosphorylation might shut down kasi nga oxygen yung ating final electron acceptor here. Okay, note that this this is where the importance of distal histidine enters the picture. Remember, sabi natin histidine or his E7 acts as a gate for oxygen. How? Remember that histidine is a bulky group. Now that bulkiness okay, that while kinetically inhibits oxygen or other molecules such as carbon monoxide and cyanide from binding ideally or perpendicularly to the heme plane. Okay, um, we'll show on this slide the importance of having an imperfect binding to the heme group, no, which by the way would seem counterintuitive because is it not the function of both hemoglobin and myoglobin to bind oxygen? Okay, so to answer that, okay, we have to go back on what we know that oxygen is not the only molecule that can bind to the heme group. So there. Now, so if you will notice in the figure, okay, on letter A, okay, if distal histidine is absent, you'll have here an ideal binding of oxygen and carbon monoxide. Now, this is gonna be very dangerous for us because in the case of oxygen, it will not be released to the tissues that need it. Okay, and in the case of carbon monoxide, we know for a fact that it has stronger affinity to the heme group than does oxygen. Okay, so it's gonna be even more harder for oxygen to kick it out. Okay, so now focus your attention naman on letter B. Okay, when you have his E7, okay, it forces carbon monoxide to bind at an angle, okay, around 120 degrees to heme due to the steric block of his E7. Consequently, okay, this provides advantage for oxygen, okay, because this lessens the carbon monoxide's affinity to 200 times. So see, can you imagine ang laki ng ibinaba niya? Okay, so this structure guards in the possibility of having traces of carbon monoxide, okay, carbon monoxide produced during metabolism. Okay, no, during metabolism to occupy all of the oxygen binding site on hemes. No? So, nevertheless, okay, note that carbon monoxide is poisonous in high concentration. Let's move to letter C. It is worth remembering yung sinabi natin kanina na although it's the job of myoglobin and hemoglobin to bind oxygen, it would be very dangerous for us, okay, if heme will not let go of its carried oxygen. Because, remember, your oxidative phosphorylation, paulit-ulit nating sinasabi, needs oxygen as its final electron acceptor. And if oxygen is not available, then the whole process of oxidative phosphorylation will shut down. And eventually, okay, every metabolic pathway will follow suit. So therefore, two perfect binding of oxygen to heme would defeat the purpose of having a protein that transports oxygen. So, di ba, nakikita natin na napakaganda at galing ng disenyo ng ating katawan. Okay, so this graph shows you the binding curve of myoglobin shown in light blue and hemoglobin naman shown in purple with that of oxygen. So again, it's the binding curve ng myoglobin and hemoglobin with that of oxygen. So if you analyze the graph further, it tells you that when you plot the degree of saturation, okay, of myoglobin, again shown in light blue, against oxygen pressure, okay, you will observe a steady rise until you reach complete saturation. And then at that point, the curve levels off. Now, the binding curve of myoglobin is said to be hyperbolic. Okay. On the other hand, okay, the shape of the binding curve of hemoglobin or hemoglobin, okay, again shown in purple, is said to be sigmoidal. Okay, if you also plot, it's said to be sigmoidal, okay, if you also plot its degree of saturation against oxygen pressure. Now, the reason for that, the reason for it being sigmoidal, no, is because of cooperative binding as what we have mentioned earlier. By the way, if I have not mentioned it pa, kay cooperative binding is only present in hemoglobin, no? So it's absent in myoglobin. Okay, so kaya ganyan yung graph nila. So, however, note here that even though there is cooperative binding in the case of hemoglobin, no, its binding curve is still lower than that of myoglobin at any pressure. Okay. So this means that an oxygen pressure, so this means that at any oxygen pressure, okay, myoglobin will always have a higher percentage of saturation than that of hemoglobin. So there, si myoglobin ang nagwagi, no? So siya ang nag-uwi ng corona. So, kidding aside, okay, in other words, myoglobin can be fully saturated with oxygen in the tissues, while hemoglobin naman requires much higher oxygen tension to become fully saturated, no, which only occurs in the lungs. Okay, so also you can also see now from the figure the fetal hemoglobin has higher affinity to oxygen. So, this ensures no, that nasusupply yung oxygen na need ng fetus. Okay? So, now we move to deoxy and oxygenated hemoglobin. So, basically, when you say deoxygenated or deoxyhemoglobin, oxygen has been removed or simply there's no oxygen bound to the heme group, no? Or konti yung oxygen na bound to the heme group, while oxygenated is the reverse. Okay, so if you will notice in the figure, okay, when hemoglobin is deoxygenated, the heme is domed, okay, and has a nonpolar configuration. Note that this is characteristic of a deoxygenated heme group. Now, the reason for it being like this is because the valence electrons in the atoms, surrounding the heme group and the valence electrons in the histidine residue, particularly the proximal histidine, okay, form electron clouds. And since electrons repel one another because they have the same charge, okay, the regions occupied by the valence electron of the heme group and that of the histidine residue, K, are pushed apart. Hence, the porphyrin here adopts a domed nonpolar configuration. And as you can see, iron is out of the plane of the porphyrin ring. Okay. However naman, okay, when iron binds oxygen, meaning the hemoglobin is oxygenated, okay, the porphyrin ring adopts a planar configuration because, okay, because the iron is also linked with the proximal histidine. No? So binding of oxygen would result on the movement of the globin chains. No? So by the way, I think I was not able to mention this. Globin chains refer to the monomers present in hemoglobin. So yung alpha 2, beta 2. So now movement alters the interface between the alpha and beta dimers because that movement draws the his F8 closer to the heme group. Hence, the iron now lies in the plane of porphyrin ring. Okay. So note here that those shape change in the heme group, has important implications for the rest of hemoglobin. Same thing when the iron atom moves into the porphyrin ring upon oxygenation. Now remember, the movement of histidine F8 shifts the position of other amino acids that are near the histidine. Now, when amino acids in hemoglobin are shifted due to oxygenation, okay, the structure of the interfaces between the four monomers are altered. Okay, so this is the reason why when a single heme group binds oxygen, the whole protein changes its shape such that, okay, it becomes easier for the other three monomers to bind oxygen. No? So if you're getting at what I'm saying here, this is the cooperative binding. By the way, these are the two states of hemoglobin. No? So deoxyhemoglobin or the T state, which stands for taut, okay, it's so taut that it doesn't want to bind oxygen. And if hemoglobin is in the lungs, okay, and there's very high concentration of oxygen, that volume or partial pressure of oxygen will force one of the O2 molecules to bind. Remember the oxygen tension that we mentioned earlier. Okay, this puts the hemoglobin into the oxyhemoglobin or R state, now which stands for relax. Okay, wherein it's so relaxed, okay, that it doesn't mind binding oxygen into one of the heme group and positively, or and positive cooperativity takes effect. So, and then also releasing it.